So I hope what I'd like to do this morning is introduce you to a concept which maybe Nir already introduced you to, this trade-off between living healthy and living long, and then give you some perspective as to whether these two components of human health can come together. So this is my disclaimer. I am a founder of Unity Biotechnology, which is developing pharmaceutical interventions into the process I'm going to talk to you about, but I'm not going to talk about anything having to do with the company. So let me start with, you all know this, because this is your job, right? That population structures are changing. So this is the population structure of the US in 1950, and this is the 70-year mark, and you can see that most of the people were young, and there were very few people who were old, very, very few people who were living over 100. And this is, of course, the projection for 2030. So what is amazing is the amazing growth of people over the age of 70, and also the real expansion of the number of people who are living beyond 100 years of age. So I would say this is good news. Our species, is living longer. I think everyone would agree, maybe? Yeah? No? Not if you're an insurance company. I know, it's okay. Okay, but here's the bad news. The bad news is we cannot afford it. I mean, this is the healthcare, this is actually an old slide, and this is the average healthcare cost for the last 2 years of life with increasing age. And you can see there is just this exponential rise in healthcare costs as people get older. And as we now see this burgeoning population of older people, this is really going to break the bank. I honestly don't understand why politicians are not more upset and alarmed about this, but this is really going to put our national debt way beyond our means. So the question is, why has this happened? How did it happen that we, Homo sapiens, have been able to extend our lifespan, our average lifespan, at such an amazing cost, at such a detrimental cost? And these are still questions. I will not pretend to provide you with clear answers, but I will try to put in context what we, meaning we people who study the biology of aging, think has happened. And then hopefully I won't depress you too much, and then I'll be able to tell you what we think we might be able to do about it. So why are we living longer? And why is that triumph draining our resources? So this is really the question, and the answer is obviously clear, and that is nobody dies of good health. We die of diseases. And what I've listed here is just a partial list of the many diseases of aging that are chronic conditions, and they're very expensive to treat. They're expensive to treat because they persist over a long period of time. They're expensive to treat because they often require constant treatment. And of course, they're expensive to treat because the population that's being treated for these diseases are somewhat frail. Comorbidity, as you know, is a big problem in the elderly. So think about these diseases of aging. And they're very different cell types, so your brain, your heart, whether you can hear or see well, how your muscles work. So we're talking about very different types of tissues, very different cell types, and yet all of these diseases have something in common, despite the fact that they affect very different parts of your body, and they have very different effects on the quality of your life. And what they have in common is that all of these diseases have the same trajectory over the lifespan. And by that I mean all of those diseases are extremely rare in young people, and somewhere around the midpoint of the lifespan, so 50 to 60 years of age in the case of humans, all of these diseases rise with approximately exponential kinetics. Now, you can imagine two possibilities. One is this is just an incredible coincidence that your heart, your lungs, your kidneys, your liver, they all begin to fall apart at the same time, but for totally unrelated reasons. People who work in the biology of aging nowadays really don't believe that anymore. We really believe that there are some fundamental processes, which for lack of a better word, we call basic aging processes, and that it's these processes that are driving these very, very different types of diseases. So this is still a hypothesis, I should point out. We haven't really totally proven that this is the case, but I would say the preponderance of evidence suggests that something like this is going on, that there are a few fundamental processes that's driving the deterioration of all of these different types of cells and tissues in your body. So that still doesn't beg the question, though, or answer the question as to why it is that we're living longer and yet we have this enormous burden of the healthcare costs. And the answer to that lies in really what modern medicine has achieved in the past, say, several decades. So the good news is modern medicine has got to be pretty good right now at treating individual diseases. If you have high blood pressure, there are drugs to control that. If you have high glucose, there are drugs to control that. If you have poor muscle, there are exercise regimens that we know will help postpone sarcopenia. So medicine has become very good at identifying pathology and then devising ways to mitigate that pathology. What we have not done, we, the biomedical research community, what we have not done is we have not found ways to attack those fundamental aging processes that we think are driving all of these diseases. So that is still something that is on the horizon. But if we're correct that there are basic processes that can be targeted, then we will absolutely revolutionize medicine. Medicine will no longer be practiced the way it is in what has been called a silo approach, meaning something's wrong with your heart, you go to your cardiologist, and your cardiologist is not going to really ask whether you have macular degeneration, and vice versa. And this goes on for all of those diseases that I mentioned to begin with. So what modern medicine really needs to do is to think about aging as a process that drives multiple diseases, and if that happens, then there will be a great need for what we call gerontologists or geriatricians, people who will be studying the aging process and treating your aging as opposed to treating your macular degeneration or your failed kidneys, et cetera, et cetera. So this is still a dream, but it is something that most of us have some confidence is on the horizon. It's hard to say when, but we think it's there. So I'm going to try this morning to answer these three questions for you, at least to the best of current knowledge. And again, I want to emphasize, we really don't have all the answers. There's still lots of research that needs to be done. So the first is, if it's true that there's a basic aging process that's driving all these different diseases, how do you identify it? How do you go about deciding what that basic aging process is? Because you have to know what it is before you can dream of intervening. The second question is, can we intervene? Maybe these processes are so fundamental to life. For example, if we stopped breathing oxygen, we would have no oxidative stress, and we would probably do away with some segment of aging. Well, that's not very practical. It's not going to happen. So is it going to be feasible to target a fundamental aging process in a way that's going to be translated into at least increased health span? And then the last question is, will this intervention, if it did exist, have any effect on maximum lifespan? So let me try to go through these questions and start with this question of how do you go about identifying a fundamental aging process? Now, there are probably lots of ways. I mean, scientists, especially biologists, are very opinionated about the way things should be done, should be done. So what I'm going to tell you is sort of how my lab went about tackling this problem. And I just want to emphasize there are lots of other ways to think about the problem, but we sort of went back to this schematic here. And we thought about these diseases of aging, and we realized that most of these diseases are degenerative. And by degenerative, I mean the tissue doesn't work as well or the cells don't work as well. So you can consider them loss of function diseases. Whatever target organ or tissue you're studying, it's not working as well as it should. But there's one outlier in this scheme. You can guess which one it is, right? It's right here. Cancer. I would be very hard-pressed to call cancer a degenerative disease. If you think about what a cancer cell needs to do in order to form a disease that will kill you, that cancer cell has to acquire new properties. So in a way, you could say, well, most of aging is a loss of function disease, but then there's this thing called cancer, which is kind of a gain of function disease. And maybe instead of asking the very, very broad question of how do we identify a basic aging process, we can simplify the process a little bit and ask this question. Is there a common biology that links the degenerative diseases of aging to cancer? So that's the question that we asked. And I was actually trained as a cancer biologist, and so we started with the question of what do we know about cancer? Now, cancer research is older than aging research, at least modern cancer research, and so the cancer biologists are way ahead of the aging biologists. People who work on cancer, we know what causes cancer. And the first thing we know is that it's caused by mutations. And I don't mean mutations that you're born with. I mean mutations that you acquire in your cells as an adult. So these are the conversion of what's been called proto-oncogenes into oncogenes, or the loss of tumor suppressor genes that occur after birth and have nothing to do with your genetic susceptibility. So most cancers are not caused by genetic susceptibility, but rather is caused by somatic mutations, mutations that you acquire in the soma of your body as an adult. So we've known that for years. But we also have known that's not enough. And we've known that even longer than we knew that mutations are the drivers behind cancer. And these go back to experiments that were done in like the 40s and 50s. I think the pioneer in this was a woman named Beatrice Mintz. So she was studying cancer. So she had a mouse had a tumor. She took the tumor out of the mouse, chopped it up, put it in culture, grew up those cancer cells, and then she divided them into two. One group of cancer cells she put into the mouse in a certain tissue, say the liver. Mouse got a horrible tumor, died of cancer, no surprise. The other half, she took those cells and she put them into a different tissue, like the kidney, and nothing happened. The mouse was fine. And that was the first hint that mutations are probably not sufficient. You need something else in order to get that mutant cell to express itself and begin to develop into a lethal tumor. And what that something else now we know, due to pioneering work that's been going on for the last couple of decades, is you need a tissue environment that is conducive to those mutations expressing themselves. So here's an example. This is work from a colleague of mine, Mina Bissell, who is at the Lawrence Berkeley Lab. So what you're looking at is a organization of several cells from the breast. So these are normal human breast cells. They're put into a three-dimensional culture, sort of a gel. And those cells will organize themselves into alveoli that look very much like you would see if you sectioned through a normal human breast. So here they are. The red are the nuclei, and the green is this protein that helps the cells organize into this structure. This is breast cancer. So you can see it's a mess. The nuclei are lobulated. This protein, this green protein, is all over the place. More importantly, the cells are not organized. They're invading into this three-dimensional culture. This is typically what cancer cells do. But now this panel here is the same cancer cells, but they were tricked with an antibody into thinking they're in a more normal tissue environment. And you can see what's happened. They don't invade, their nuclei are better organized, green protein is in the right place. And this is really one of the first demonstrations that you can take a very messed up, mutated cancer cell and trick it into becoming more or less normal. These are not completely normal, but more or less normal by changing the tissue environment. So, there's a bit of a dilemma in what I just told you. So I told you you need now two things to form a cancer. You need mutations, and you need a tissue environment that is permissive for those mutant cells to begin to grow into a tumor. So here's the problem. Everybody in this room, I guarantee you, 100%, you have mutant cells in your body already. Actually, if I were talking to a class of high school students, I would make the same statement. So we now know that those potentially cancer-causing mutations begin to arise very early in life. Actually, as soon as you can begin to measure them. So even in utero, you can begin to see a few cells that are picking up these mutations. The reason is the best time to form a mutation is during cell division, because you have to replicate the genome, 3 billion base pairs, you have to make it perfect with no mistakes, and it's pretty good, but it's not perfect. So you begin to acquire mutations very early in life. That's one problem. Second problem is that tissue structure begins to change long before that midpoint of the lifespan. So, for example, if you took a liver biopsy from a 15-year-old and a 50-year-old, gave it to a pathologist blinded, pathologist will instantly tell you who's young, who's old, just by looking at the structure of the tissue. So, the two things you need for cancer are mutations and a change in tissue structure. And I've just told you they start really early in life. So the question is, why is it that on average, we don't get cancer for the first 50 or 60 years of our life? Remember that curve I showed you? Cancer falls right on that line. Very rare in young people, and then starting around the midpoint of the lifespan, is an exponential rise in the chances of developing cancer. So what's going on? Why are we so protected early in life and not protected later in life? So again, the cancer biologists, being way ahead of us, they solved that problem. They know the answer. And the answer is that we have encoded in our genome 100 or more genes that are called tumor suppressor genes, and they converge on a few fundamental mechanisms, cellular mechanisms, and their job is to suppress the ability of cells to undergo the process of becoming a tumor cell. And they work pretty well. They work for 5 or 6 decades, and then the system begins to fail. So I'm going to argue now that this remarkable protection we have against developing cancer for half of our lifespan came at a cost, an evolutionary cost. And that cost is what we call aging. So let me explain to you why we think this hypothesis is worth, and it is a hypothesis, why we think it's worth considering, and it will have a big impact in how we consider the future of longevity research. So maybe 20 years ago, two very famous cancer biologists, Kinsler and Vogelstein, proposed that it would be very useful to think about these tumor suppressor genes as coming in 2 basic flavors. And they called them caretakers and gatekeepers. So caretakers are very straightforward. Caretakers are genes that code for proteins that take care of our genome. So these are things like DNA repair genes, antioxidant defense genes. They basically are good guys, and you can even consider them longevity assurance genes. They help assure our longevity by taking care of our genome. Very straightforward. The second group is not so straightforward. They call these guys the gatekeepers. And what gatekeeper proteins do is they tend to be sensors of cell state. And what they sense is whether a cell is damaged or whether a cell is stressed. And if they sense that that damage or stress puts the cell in danger of becoming cancerous, it gives that cell two options. And the two options are to die or to never divide again. So cell death is called apoptosis. This is also called programmed cell death. And the other process is called cellular senescence, which means the cells absolutely forever lose the ability to divide. So it's easy to see why these gatekeepers are tumor suppressors, right? A dead cell cannot form a tumor. A cell that can't divide also cannot form a tumor. So these are great things to have. I should tell you, we now have mouse models, transgenic mouse models, where we cripple the ability of the cells to undergo cell death or cripple their ability to undergo senescence. There are even people who are born with rare mutations, so this is quite rare, but still, they're born with mutations that do either of those things, and we know what happens to those mice and those people. They die an early death due to cancer. So we know for sure these processes prevent cancer. That's good, right? Think about now 50 years of living in a body where cells are slowly but constantly undergoing cell death and cell senescence, losing the ability to live and losing the ability to divide. So in the first case, what's going to happen is those cells disappear. Stem cells will then be mobilized to replace that dead cell. And as a consequence, there's a constant draw on those stem cells to replace the dead cells. And eventually, you run out of stem cells. So the tissue undergoes atrophy or degeneration. The same thing happens when a tissue begins to fill up with non-dividing cells. The tissue has to maintain a certain size, and if that tissue doesn't have very many dividing cells in it, then its ability to regenerate and repair becomes impaired. And I'll show you that this process of senescence is much more complicated than the process of cell death, and that senescent cells can even affect their neighboring cells in a way that dead cells cannot. So think about these phenotypes. This is what we call aging. This is aging. This is tissue degeneration. And I'll even show you that in some cases, senescent cells, which we designed to prevent cancer, can late in life begin to drive cancer. So let me show you one other reason why this is an extremely important concept in thinking about aging and longevity. Because this idea that there can be forces that are protective and beneficial when you're young, but detrimental when you're old, has been around since the 1950s. It's a process that has been termed antagonistic pleiotropy, and it was first formulated by a very well-known evolutionary biologist called George Williams. So here's the idea. Let's say you start off with a bunch of organisms. Let's say people and mice. You start off, they're all young, they're all vigorous, 100% of them are alive, and then you follow them, just the way many of you do in your work, and you look at the decline in the population. So for 99.99% of our evolutionary history, that decline looked something like this. And when zero population was reached, nobody died of aging. Didn't happen. People died mice died primarily of infection, but also predation, loss of food, loss of control over the climate. So this is a natural environment in which most of our evolutionary history was dominated by this kind of environment. And so if you look at what these ages really are, in the case of humans, there were very few humans who lived beyond the age of 50. Actually, there were always outliers, and there still are outliers. But for the vast majority of people, there were no elderly. Everyone was dead, primarily of these extrinsic hazards. Same thing is true of mice. The average mouse out in the field lives 4 to 6 months. Same thing, they die of infection, they die of predation, cats and hawks, not enough food, they get too cold or they get too hot. So, in the last few decades, our species, Homo sapiens, we have done an amazing thing. We have changed our environment from what has been called a natural environment, high hazard, to a low hazard, protected environment. Now, of course, I'm talking mostly about the developed world, but for the most part, we have conquered the major infections that shortened our lifespan. We have control over our food supply, control over our climate, and so far as I can tell, our biggest predators right now are ourselves. So we are now living way beyond our evolutionarily less fit ancestors. And we've done this for our mice. We take these mice, we put them in an animal colony, no cats, no hawks, lots of food. And mice will live 3 to 4 years if they're protected in the environment that we create in our animal houses. So this is amazing when you think about it, what we have done, we Homo sapiens have done. But of course, it came at a cost. And the cost is that there was no evolutionary pressure to improve anything that happened to keep these organisms alive if it was detrimental way out here. And that's the concept of antagonistic pleiotropy. And it really can be paraphrased by saying there are things that are good for you when you're young, and those very same things with you are going to be bad for you when you're old. And I'm going to make the argument that cellular senescence is one example of something that can drive aging, but only later in life. And let me explain to you why we think that's the case. So first, let me explain to you what happens when a cell becomes senescent, when it loses this ability to divide. So we think of senescence as a kind of stress response. Cells are damaged or they're stressed. The gatekeeper, tumor suppressors say, okay, guy, you either die or you senesce. And actually, by the way, I should tell you, we still have lots of questions about how that decision is made, whether to die or to senesce. Still a big unknown. But nonetheless, the cell undergoes senescence, and 2 things happen. So I told you about one. I told you that the cells stop dividing. But something else happens. The cells also begin to secrete molecules that can have a profound effect on neighboring cells. And that turns out to be the key to understanding how all these different types of stresses, from things that damage the genome to oncogenes, activation of oncogenes, for example, many cells will undergo senescence when an oncogene becomes activated. And what happens is these stresses then induce these phenotypes, the loss of growth and the secretory phenotype spewing this stuff out, and they're linked. They're absolutely linked. So we don't yet know of ways to separate them. And so as a consequence, this is a balancing act. The good news is it's preventing cancer for many years, but the bad news is it's driving those phenotypes, and I'll show you evidence it's driving some of the diseases that we know, that we call age-related diseases. So how do we know this? Well, we know this now because in the past 20 years, many laboratories have worked on this problem, and we now have markers, biomarkers, where we can ask when and where do senescent cells appear. So I've listed a whole bunch of these markers here. I should say one of the problems in the field is we have no specific marker, so we have to look at multiple markers, but nonetheless, we can do that and have some confidence that we can identify senescent cells. And I just want to point out the key features of a senescent cell. So the first is this inability to divide. Cell will never divide again. The second is it expresses a protein called P16. I'm going to come back to P16 in a moment. The reason why P16 is important is it's a tumor suppressor gene, And it's responsible for stopping the cells from dividing. It stops the cell cycle. But the third thing that is important about senescent cells is they don't like to die. So I'll show you that we have found ways now to get them to die, but for the most part, these cells are stable. They exist, they stop dividing, they exist, and they don't have the good grace to go away. So using these markers now, many labs have asked the question, okay, when and where do you find these cells in vivo, in the body? And the first thing we know is they increase with age. And this is true for virtually every vertebrate species, from humans all the way down to zebrafish, that have been looked at. Multiple tissues. As we age, these cells, remember, they don't die, they become senescent, they're protecting us from cancer, but they're slowly building up over time. But the second place we see them is at increased number for many, many of those age-related diseases that I showed you several slides back. So here are some examples. This is COPD, chronic obstructive pulmonary disorder. This is the major cause of lung failure in the elderly. And what you're looking at is markers of senescence in the smooth muscle cells of the arteries in the lung. And you can see, if you look at age-matched controls, the diseased lung has more of those senescent cells than age-matched non-diseased lung. Next example I'm showing you is a brain from an Alzheimer's patient. So if you compare age-matched Alzheimer's patient brain with normal brain, what you see are more senescent cells in the Alzheimer's brain. Now, the cells that are senescent are not the neurons. This may come as a surprise to some of you, but although neurons are, of course, what make the brain work, they're not the most abundant cell in your brain. The most abundant cell are a cell type called astrocytes. Astrocytes have the ability to divide, neurons do not. And brain cancers are caused by astrocytes, not by neurons. So again, it's the dividing cells in the brain that are in danger of becoming cancerous, and they undergo senescence. And there are more of them in a diseased brain than a non-diseased brain. So I know this is a very American slide. I'm embarrassed when I show this in Europe, but it's true. You can say senescent cells are a smoking gun, right? They are present at the right time and the right place to be driving multiple age-related pathologies. They're all over the body. They increase with age. But the question is, how? Why would these cells be causing these very, very different types of diseases? And the second question is, do they actually do it? Everything I've told you so far is correlation, no proof. So I think now what I'd like to do is first explain to you how, but then I'm going to get to the question of, do they actually do it? Obviously, the answer to this we now think is yes, otherwise I probably wouldn't be here. But the yes is in a mouse, so we still don't know in people. But for sure, I think we can say this is true in the mouse. So how? You have these cells that are not dividing, they're sitting there. Why are they driving all these different kinds of diseases? And the answer has to do with what they're secreting. So we tend to look at these secretions as coming in two basic flavors, and I won't go into the details, but the main point is whatever they're secreting has the ability to cause a process called inflammation. And what we know about inflammation is that it is the hallmark of aging tissues. All aging tissues tend to show this increase in inflammation. We know that inflammation can destroy tissues. We know that it can disrupt normal tissue functions, so the tissue just doesn't work as well. We know that it can prevent stem cells from functioning. And we also know that inflammation is a major risk factor for the development of cancer. So let me just explain a little bit more about inflammation. So you know when you cut yourself, it gets bright red. This is acute inflammation. You will die if that process doesn't happen. The whole idea behind an acute inflammatory response is to get the tissue to begin to repair and regenerate as quickly as possible and to fight off infection. And the way inflammation works is that the damaged cells secrete molecules, and those molecules do two things. They attract certain immune cells, and these are, many of you may know this, the immune system has two basic flavors, the innate immune system and the adaptive immune system. So the innate immune system is ancient. It's present in worms and flies and humans. And the whole idea behind an innate immune system is those cells come in and they try to kill anything that looks foreign non-specifically. And they do it by making damaging molecules. They make bleach, they make nitric oxide, they make hydrogen peroxide, and they do it non-discriminately. Takes time then for the adaptive immune system, the intelligent part of your immune system, to then be more specific and attack those specific bacteria or viruses. So what chronic inflammation is a low-level infiltration of those damaging immune cells. And in the meantime, those factors that are attracting those immune cells are causing the cells that are nearby to change in their function, to make them become less specialized and less functional. So when I say that there's inflammation caused by senescent cells, it's a double danger to the tissue. The tissue is being damaged by the immune system, and the tissue is losing function because of those secreted molecules. And of course, every one of those diseases I showed you on that earlier slide is either caused or greatly exacerbated by this condition of chronic inflammation. And that includes everything from neurodegeneration to cancer. So we think this is how this happens. The second question, though, is do senescent cells actually do this? So this is still hypothetical. And the answer we really think is yes. So this secretory phenotype, we and many other labs have worked on this over the course of the last 20 years, have shown that the secretions of senescent cells have profound effects on their neighbors. And I'm going to give you 3 examples. So the first example is back to the mammary gland. We can take now breast cells, normal breast cells, grow them in these 3-dimensional gels where they form these beautiful alveoli. And one thing I didn't tell you in that earlier slide is if you add hormones, the hormones that a woman produces when she's lactating, if you add that to the cultures, those cells will now produce milk. So in this case, the red staining is a milk protein. So this is functional and morphological differentiation of those breast cells. So if we embed in these cultures non-senescent cells of any type, we still get these beautiful alveoli, lots of milk production. But if we embed in these cultures senescent cells, you can see what happens. First of all, lots of ugly alveoli. They're no longer formed these beautiful round shapes, and milk production falls. So the function and the structure are degraded. This work was done by a graduate student in my lab called Simona Paranello, and she was able to even identify the factors that are responsible for this decline in structure and function. So we have some idea of what the cells are producing, and we can neutralize those factors and prevent this from happening. Now, the second example I'm going to show you has to do with stem cells. So what you're looking at is a micrograph of human neural stem cells. So these are the stem cells in your brain, and they have to do 2 things. They have to proliferate when there's an injury, and they have to migrate to the site of the injury. And we can measure that in culture. So here, the green staining is the ability of the cells to proliferate. And you can see if we take conditioned medium from astrocytes, those brain cells that decline, that become senescent in the diseased brain, if we take conditioned medium from those cells and put them on these neural stem cells, if they're non-senescent, we get lots of DNA replication, lots of cell division, and that falls when the conditioned medium is from senescent astrocytes. And the same thing is true as the ability to migrate. This is just the ability of cells in a wound to migrate and close the wound, and you can see that that's retarded by something that those senescent astrocytes are making. We don't know what that factor is. We're still working on that. The last example I'm going to show you really shocked us when we first discovered this. It was first discovered by Anna Kratolika in my lab, and then it was followed up by Jean-Philippe Coppet. So what you're looking at here is tumor size over time. And what we've done is we've injected into the mouse mammary gland pre-malignant breast cells. So these are not breast cancer. They're pre-malignant. They have some mutations, but not enough to form a tumor. So you can see over 6 months, no tumors. But now, if we co-inject another cell type that's either not senescent or senescent, you can see that the senescent cells will cause those pre-malignant cells to transform to full-blown tumorigenicity. These are terrible tumors. We have to sacrifice the animals because they grow so fast and they're so big. This is an example of human breast cancer cells injected into a mouse breast. And then 6 weeks later, we just excise the tumor. So this is with no co-injection of other cells. This is with non-senescent cells. And this is what happens with senescent cells. These tumors are not only larger, but they have more blood vessels. More blood vessels mean those cells are going to metastasize. So the idea is that over time, we accumulate senescent cells as we age. And it's approximately exponential. And those senescent cells then secrete molecules that cause their neighbors to fail to function. And that drives, we think, the degenerative diseases of aging. But it's even worse than that, because I told you, with time, we're also accumulating mutations, so-called pre-malignant cells, and we're arguing that senescent cells will now cause these pre-malignant cells to convert to be full-blown cancers, and this may be one of the reasons why cancers go up after around the age 50, and they rise exponentially with age. So, if you're not depressed, you haven't been paying attention. So I told you we need this tumor suppressive mechanism, otherwise we die an early death due to cancer. Now I'm telling you, you're going to get cancer anyway and you're going to die. So what do we do about this? But I think what this body of evidence really suggests is that probably the senescence process is a fundamental aging process, and it probably is driving multiple age-related pathologies. So now the question is, can we do anything about this? Because this is an evolutionary trade-off. We can't prevent cells from becoming senescent. We have to live with it. But how do we now get around this late-life deleterious effects of these cells? So we imagine there might be two solutions to this problem of trying to live with senescence but not suffer as a consequence of it. And the two solutions were, one, what if we could get the cells to stop secreting? Just give them a drug that maybe turns off secretion. So now they're no longer making all this stuff. That's one possibility. And the other possibility is, what if we could get those cells to become senescent but then die? So I'm going to tell you right now, I'm not going to spend much time on the first strategy. We spent a lot of time working on this. The good news is we were able to identify why the cells are secreting. We were able to identify the pathways that drive those secretions. The good thing about those pathways is they're driven by proteins that are targets of drugs. And the really good news is that there were already drugs in the clinic that were targeting those pathways. So here are the three pathways. One is the DNA damage response pathway. The other is this stress-activated pathway that drives the inflammation. And the third is this longevity pathway, which is the target of rapamycin. So that's the good news. We have the pathways, we have the protein targets, and there are drugs out there. Here's the bad news. You cannot live with these pathways being constantly suppressed. I mean, obviously, you can't suppress your DNA damage response for any long period of time. That would put you at great danger for developing more mutations, et cetera, et cetera. So we were great to figure out all this biology, but we didn't think it was a very practical issue. And the other problem with these pathways is you need constant pressure from the drug. As soon as you take the drug away, the cells are still there, they start secreting. So drug companies hate when I say this, but it's true. There is really no such thing as a perfectly safe drug, and as a consequence, we thought the better thing would be to try to find a way to simply get the cells to die, to become senescent and then go away. So I'm going to tell you now, this is all still very relatively new work, just in the past 5 years or so, that we can now do this in the mouse. And we can show that indeed senescent cells drive multiple pathologies, and then I'll show you what happens to lifespan. So the first study was actually done by our colleagues at the Mayo Clinic. This is from Jan van Dersen and Jim Kirkland's lab. And what they did was they devised a transgene. So this was a foreign piece of DNA put into the mouse genome. I'll explain to you what that gene looks like in a moment. But the main thing about this foreign gene is that that gene would cause certain cells to die when you give a drug that has no effect on non-senescent cells. So the drug only affects senescent cells expressing this foreign gene. So this is not ready for prime time in people because we can't make transgenic people. But still, we make these transgenic mice, we give them a drug, senescent cells die. So a mouse lives 3 to 4 years. They didn't want to wait that long. So what they did was they crossed that transgene into a prematurely aging mouse. And that's what you're looking at here. This is a prematurely aging mouse. It lives only about 9 months. And it dies of multiple things, but mainly heart problems. So here's this mouse, and it's got lots of senescent cells. Here's its litter mate, and it has almost no senescent cells because they were given this drug that causes the transgene to cause senescent cells to die. So you can see the difference. This guy has cataracts, sarcopenia, so wasted muscles, osteoporosis, has almost no subcutaneous fat. And here's its litter mate. It has no cataracts, no sarcopenia, no osteoporosis, lots of subcutaneous fat. Clearly, this mouse is healthier than this mouse. Unfortunately, they all died at 9 months of age. So this mouse also died at 9 months of age, and it died of the heart problems. But this was the first demonstration that senescent cells do indeed, at least in the mouse, drive these age-related pathologies. So here's a little bit of details about the transgene. This is our mouse. Our mouse is very similar to the Van Dersen mouse. This P16 tumor suppressor that I told you senescent cells express, we took the elements that turn that gene on, and instead of turning on the normal P16 gene, we now have it turning on this totally foreign gene. What this foreign gene does is it allows us to visualize senescent cells in a living animal, to be able to isolate senescent cells from tissues of that animal, but more importantly, it enables us to kill those senescent cells, and only senescent cells, by giving a foreign drug. In this case, it's a drug called ganciclovir. It's actually an antiviral drug. And the gene that causes this antiviral drug to become a toxic moiety is a viral protein. So using this mouse now, we can ask lots of questions. So the first thing is, if we take an old mouse and we give it ganciclovir, the drug that kills senescent cells, can we eliminate those cells in the body? And the answer is yes. So here we visualize the senescent cells by this process called luminescence, causes the mouse to glow. And you can see that they glow more and more with age. And then if we, at 23 months of age, we give them this drug, they don't glow anymore. And this is one of those markers of senescence. It's an enzyme. It turns senescent cells blue. And this is visceral fat. So unfortunately, visceral fat, belly fat, has lots of senescent cells. So you can see very little in young animals, lots in old animals. We give the animals this drug, and the senescent cells are gone. So this is very good. So now we've used this mouse, and the Van Dersen lab has used their mouse, and we looked at lots of age-related diseases. We've looked at, so one of the problems with chemotherapy is it causes premature aging. This is a big clinical problem right now, and we've shown that that's due to senescent cells. We've looked in a Parkinson's disease model and shown that senescent cells can drive aspects of Parkinson's disease, aspects of osteoarthritis, aspects of atherosclerosis, and the list is growing. There are now probably 24 labs that are using these mice to look at the ability of senescent cells to drive these very, very different types of age-related pathologies. I'm going to show you data for only 2 of them because they're very graphic and they're very short stories. So the first is Parkinson's disease. You may know that there is a herbicide called paraquat. It was used actually for a long time in California. It's no longer used legally because migrant workers who were exposed to this herbicide develop prematurely Parkinsonian symptoms, so loss of motor neuron function. And so we can treat animals with mice, of course, with paraquat, and they develop Parkinson's disease. So here's what it looks like. We take the mouse and we give it paraquat, or we give it a control, just the solution in which the paraquat is dissolved in. We put these mice in a cylinder and they try to get out. And it requires motor neuron coordination. These guys have to stand up on their 2 legs and try to get out of the cylinder. When we give them paraquat, they can't do it. They just can't maintain their balance. But now if we take the paraquat-treated mice and get rid of senescent cells, which we know are the astrocytes in the brains of those animals, now they have no motor neuron problems. The second example I'll show you is osteoarthritis. This is injury-induced osteoarthritis. So what you can do is you can surgically cut a ligament in the leg of a mouse, and what happens is you get bone rubbing on bone, and that develops osteoarthritis. So now we can ask what happens in that osteoarthritic joint, and it turns out there's lots of senescent cells in that joint. Now we can get rid of them. And the best example of what happens when you get rid of them is you ask, how much weight can the animal put on that injured leg? So these are animals animals have no surgery, they put 100% of their weight on that foot. Animals that are sham operated, that we don't cut the ligament, but we just open them up, 100%. But now, when we cause the osteoarthritis, they can only use that leg about 50%. If we get rid of the senescent cells, they're back up to 100%. So the list is going to go on and on. I think there are going to be many, many more examples about how these senescent cells can drive all these pathologies. So now I want to take one step backwards. Some of you should be a little bit puzzled and say, well, you told us that there's this tumor suppressive mechanism called apoptosis. Cells die. What's great about apoptosis? No inflammation. Cells just go away. So why did this very messy, ugly second tumor suppressor mechanism evolve? And that's the question that we've been asking ourselves for several years until we discovered the answer. And the answer is that this secretory phenotype does in fact have a good function. And the good function is to promote wound healing. So let me show you a little bit of data from this. This work was done by Marco Di Maria in the lab. So what he did is he did a little punch biopsy on the back of the mice. And remember I told you we can now see senescent cells glow. So that's what you're looking at. These are the senescent cells and they're glowing on the back of the animal. So we say, okay, senescent cells are forming at these little wounds. This is only 4 days as the wounds are healing. And so we can ask, what happens to those senescent cells? And the answer is, they're only there transiently. So they come up a few days after the injury, and then they go away. We can take those senescent cells and make them die by giving them the drug. And that's what's shown here, right? Perfectly flat. And now we can ask, what happens to wound healing? And what happens was really a surprise. wound healing is worse. So here, for example, are wild-type mice that are given the drug, so they don't have the transgene, so the drug does nothing to a wild-type mouse. But the transgenic animals, so these are the senescence-free wounds, are much slower to heal their wounds, and those wounds also have more fibrosis. Now, what is really critical about this trajectory is it's very different from aging. So I showed you I didn't show you, I told you senescent cells are present in many aged tissues, including the skin. But the difference here is that during wound healing, the cells are there for just a short period of time, a few days. With aging, they're there all the time. And we think that's the critical difference. In fact, we can model that and cause those senescent cells not to die, and we can show that eventually that becomes maladaptive. So first it helps the wounds, and then it makes them worse. But the real beautiful thing about this study from Marco was because we can isolate those cells, he was able to ask, what are they making that makes them heal wounds better? And he found the factor. He found one factor, and it completely explains what the cells do that's good for wound healing. It's called PDGF-A. It's platelet-derived growth factor A. It's a natural product that we all make. And it turns out senescent cells make lots of it. And so he can take those senescence-free wounds, which are healing slowly, and just add PDGF-A to the wound. And now wound healing is perfectly normal. So this is good news. It means that we can get rid of senescent cells and maybe replace the good factors that they make. But this is going to be a recurring theme in aging, is that there's the good news and the bad news. So I just want to end with one more slide about practicalities. So we cannot make transgenic people, at least not yet. Who knows? What we'd like to do is to find pharmacological agents that can do what our transgene can do. And up until very recently, that was questionable whether we could do that. But there are now 2 labs that have discovered drugs that are capable of mimicking what our transgene can do. They're not ready for prime time. Those drugs do have some side effects, but there's an army of chemists now that are tweaking these drugs. And they're drugs actually that were made by a company called AbbVie. And these are just the references in case you're interested. So we're really confident that these drugs that can kill senescent cells, they're called senolytic drugs, they are on the horizon. I think in our lifetime we will see these drugs come into the clinic. And I feel very confident that for some age-related diseases, those drugs are going to be very beneficial, some of the examples that I showed you. But the real question is, what will this do for longevity? Are we going to really now create a species where we can live substantially longer than, say, the 115 years or 120 years that seems to be at the ceiling? It's an important question, right? So in a way, we already have the answer for the senolytic drugs, and this comes from, again, the Van Dersen lab, where they took mice, they're transgenic mice now, but what they did is they eliminated senescent cells starting at around middle age all the way to the end of the lifespan of those mice, and this is what they got. So these are males, these are females, and these are the mixed sexes, and these are 2 different strains of mice. And what you can see, the solid lines are the untreated mice as they're dying, and the lighter lines are the mice in which senescent cells were eliminated throughout the lifespan, or throughout half of the lifespan of the animals. So what has happened is median lifespan has been extended 25 to 30%. These animals, no question, are healthier. They're more active, they look better, their brains are working better, their muscles are stronger, etc. So this median lifespan. This is maximum lifespan. There's hardly any effect on maximum lifespan. So there are 2 possibilities. One is that you cannot explain all of aging by senescent cells. I firmly believe that, actually. But the other possibility is that in order for evolution to take a mouse and a human, 97% genetic identity, and create a 30-fold different in lifespan, evolution had to tweak so many genes. There's never going to be a single drug that can tweak all of those genes. And that's really illustrated by what we know from many labs who have been studying lifespan extension in model organisms. So the first is the little nematode C. elegans. C. elegans lives 2.5 weeks. And the world record right now is to extend the lifespan of that animal 10-fold. That's amazing. Now, C. elegans is a pretty simple animal. It's got about 1,000 cells. Every cell in that organism is known. It's known from the time it's a fertilized egg to the development of a full-blown organism, and the genetics have been worked out. The whole genome has been sequenced. Most of the functions of most of the genes have been known. Now, if you move up the evolutionary scale to, again, a simpler animal, but pretty much more complex than C. elegans, I should point out C. elegans has no dividing cells. All the cells are postmitotic. So this organism doesn't get cancer. But you move up the evolutionary scale to the fruit fly. This is another favorite model organism of biologists. It's actually quite a bit more complex. It has multiple tissues. And here, we have barely been able to extend the lifespan of that organism by 2-fold. And now we go to the mouse, which is, of course, infinitely more complex than a fruit fly, and we're lucky if we get 30 to 40%. So this is probably something that evolution is telling us about the difficulties it's going to be to really extend maximum lifespan. And so the Holy Grail, the practical Holy Grail for people who are working in aging, is extension of health span, being able to extend healthy years of healthy life and compress those years of morbidity. That, we think, is possible. We're not so sure, I think most of us are not so sure that it's going to be possible in any reasonable period of time to extend maximum lifespan. But of course, one of the things we've learned in biology is never say never, so we don't really know. So I'll just end with this idea that we are faced with these evolutionary trade-offs, and what we really need to be able to do is to harness the good things that some of these basic aging processes do, and maybe by eliminating senescent cells we can postpone the bad things that they do, but then we're going to have to replace the good factors because, again, there's going to be this tension between what's good for you when you're young and what we want to still be good for us when we're old. So I will stop here, and I'm just going to list, these are current people in the lab, past lab members with whom we still collaborate, and a really international group of collaborators without which none of this work would have been possible. Thank you. So we have time for questions?
Yes, thank you, Dr. Campisi. Now we'd like to open up the session for questions from the attendees. Questions, please. Please state your affiliation and your name.
My name is Tim Marr, Medical Director of Munich Re in Canada. That was a fascinating talk. Thank you very much for making something that is intrinsically very complicated understandable. I've got two questions. The first is about programmed senescence. Why has this not been like in a Darwinian way programmed out of the species? Apoptosis sounds like it's pretty good, works well to our favor, but senescence doesn't. Yet we're living with it longer and longer. That seems a little counterintuitive from a developmental perspective. My second one is, I was surprised to hear you say that senolytic drugs will be, working within our lifespan. It seems to me that the biology of aging is enormously complicated. And you made the comparison. But you said at the outset that we now understand the biology of cancer. It took ages for us to get there, and it seems to me that aging is even more complex. My sense is that is going to take an awful lot longer to understand the biology of aging. And to be able to intervene in an effective way is really a long way away. So I just wonder why you think that this is so rapidly achievable.
So let me answer the first question first, which is how did this senescence, so I want to emphasize, senescence is a program in the way that apoptosis is a program, but really why did it evolve? And the reason I gave and the reason I want to emphasize is it evolved primarily to keep us cancer-free during those years of reproductive fitness. And so it has not escaped Darwinian selection. I'm quite sure if the trend that we human beings are in right now, women having their babies later and later in life, I'm quite sure that within a few 100,000 years, evolution will have solved that problem. The problem is we don't want to wait that long. So if you wait for evolution to take care of the problem, you have to wait a very long time because evolution goes by starts and fits, and also by random selection for alleles. So I think the experiment has actually been done in fruit flies. This was done by Michael Rose, where he just simply took a batch of fruit flies and then took the ones that were the longest lived and kept breeding them. And over many, many, many generations, he eventually developed fruit fly strains that had much longer lifespans than the original strain. So we're doing it. It's just that we don't want to wait as long as it takes for evolution to work. As far as why I'm optimistic about senolytic drugs, I am not optimistic that it is going to postpone every age-related pathology. I think that's much too simplistic. And I can tell you right now, there are actually several companies that are working on those drugs. They're not thinking about using them for multiple age-related pathologies. They're going to test them for very specific pathologies. But what will happen, like metformin, I presume, is eventually they're going to be used, eventually it's going to be seen that they have beneficial effects that extend beyond the original indication. And that's going to take a long time, I think.
Geordie Postubus with Hannoveri. Thank you. That was a fascinating talk. From an evolutionary perspective, what do you think, or do we know what came first? P50, the apoptosis, or senescence? And
probably apoptosis.
Probably.
So, for example, apoptosis is very important in embryogenesis, and even C. elegans, the little nematode, which has no dividing cells in it. It was first, the genes that were first described were described in C. elegans. So it goes way back evolutionarily. Senescence was a much later evolution.
And so then I guess the next question is wound healing, as we see it now, the primary reason through antagonistic pleiotropy that we have senescence, or do we have a hint that it may be doing more?
So it's hard to answer that question because we don't know what evolution was quote-unquote thinking when it evolved senescence. But what I can tell you is that there are two laboratories, both in Spain, that have now described senescence in the embryo, including human embryos. And there, what the senescent response seems to be doing is fine-tuning the ability of certain structures to form in the embryo. So, for example, in forming digits, normally in mice and humans, you start with a petal. And then cells in between the digits die by apoptosis. Turns out that the first thing that happens in the petal is a wave of senescence at the top. And that seems to send the signal for the cells underneath to die. Now, you can make a mouse where cells don't senesce. And I told you those mice die of cancer. But they're born pretty normally. So the idea was that maybe senescence fine-tunes embryogenesis, but it's not absolutely essential. So again, we don't know which came first because we can't go back and do the experiment. But it probably has its roots during, at least as far back as mammalian evolution, or certainly the evolution of digits.
Thank you.
I'm Dale Hagstrom from Melbourne. I was just curious if you're willing to speculate or if you have a sense of what others anticipate let's look into the future, whether it's 5 years or 25 years, and when develops drugs you can use therapeutically for suppressing the senescence sort of thing. When that comes, is it likely to be most useful for people who are still, I'll call it, age 50, before they start developing all the problems, or is it equally effective for ones who've had senescence hanging around and they're 80 and they got deterioration one thing or another? Is it not likely to be very effective?
It's a very good question. The question is this going to be better at preventing age-related pathology, or can it reverse pathology? And the answer I'm going to give you is a soft answer because these are such early days. But I think the answer is going to be, it's going to be very tissue specific. So, for example, it is unlikely that if you have a diseased brain, getting rid of senescent cells after the neurons have died is probably not going to be very effective. On the other hand, the Van Dersen lab has evidence that even in an animal, these are mice in which you can force the mice to develop atherosclerotic plaques. Even in a fully formed atherosclerotic plaque, getting rid of senescent cells, which are at the base of those plaques, helps stabilize the plaques, which means that there'll be a less chance of stroke. So it's probably going to depend on the tissue and the pathology. It's definitely not going to be a magic bullet. And I'm more optimistic about prevention than I am about reversal. But these are early days. We just don't know.
Judy, terrific talk. I was surprised that somebody here called you optimistic. You ended your slide by saying nematode tenfold, flies twofold. But if you had the same slide and you would exchange the fold by additional days or weeks of life, it would be very, very different. It would be going up.
Yes.
So even, let's say, in humans, we can achieve 5% or 10%. This is huge if it's a health span. But if you could just comment about a question that was asked by the same person, that in cancer, it's so complicated. You have your genome, and then you have the cancer genome, and it's also not one genome. And the fact that aging is evolutionary conserved, that makes it really simple theoretically. I mean, don't you think that aging is a simpler a problem than treating cancers, individual cancers?
Well, first of all, I think we have to consider cancer as part of the aging process. It's not something that's totally different, I don't think. I think of all the age-related diseases, though, cancer is going to be the hardest nut to crack. And the reason is because for the degenerative diseases, there is hope that stem cell therapies will be beneficial. We're not there yet, but even in the case of the brain, there's some evidence that you may be able to rejuvenate stem cells and reverse some of the age-related deficits in brain function. Every time a stem cell divides, it has to replicate 3 billion base pairs of DNA, and it's not a perfect system, and so accumulating mutations is going to be an ongoing problem. So I think cancer is going to be a very hard nut to crack. If anything, it's going to be more complicated than aging because it's evolving. I mean, unlike many age-related pathologies which have a more or less linear decline, with cancers, that genome is constantly evolving. And until we can kill every cancer cell, the chances of curing cancer are really dire.
Hi, my name is Magali Barbieri, and I'm a researcher at UC Berkeley. You started your talk by saying that most cancers are not driven by genetics, but you also said that there is a strong relationship between cancer and aging or senescence. And I understand that part of senescence is probably accelerated by some behavior or environmental factors, but we also know that there is a strong relationship between there are large variations or significant variations within a population in the process of aging. Some people age faster than other people. And we know that there are clusters of families which seem to belong either to the fast-aging groups or the slow-aging groups. And so my question is, isn't there a fundamental genetic component? And how much weight would you give to the genetic versus the environment?
Yep. So let me first make a disclaimer. I never meant to imply that there was no genetic component to aging. What I meant to imply, or what I meant to say, was that in the development of cancer, these are the cancers develop from mutations that you acquire as an adult. And what your genetic background can set the stage for how quickly or how well you repair those mutations or reverse those mutations, but in the end, it's those mutations you acquire as an adult that drive cancer. But you're absolutely right. There are genetic loci where certain flavors seem to predispose to longevity and certain flavors seem not to predispose to longevity, and environment does play an important role. What we don't know, what we don't understand very well right now, is if you have, for example, certain genetic loci that predispose to longevity, is that because you have fewer senescent cells? It may not be. It may be something totally different. It may have to do with how well you protect against oxidative stress or how well your stem cells can function. So I really don't mean to imply that obviously if all of aging could be explained by senescent cells, those mice would have had increased longevity, and they really didn't. They had an increased median lifespan, but maximum lifespan was unaffected, and they were genetically homogeneous.
I'm Dan Bailey. Thank you. This was fascinating. I'm interested in how evolution works and all this, and whether all living beings, all the way down the phylogenetic chart, all beings who are subject to mitosis, say, are subject to senescence as you describe it? And if so, as you go up the phylogenetic chart, is it more biologically sophisticated? And I realize it could be billions of years before physical evolution creates a solution to this, but we're the species that has cultural evolution, and this is basically the fix. So can you describe with humans if that's a more advanced process than with, say, plants?
So it's a great question. What I can tell you is that senescent cells, using those markers that I described, have been found in every vertebrate animal that has been examined. And that list is not huge. That list goes from humans to rodents of various sorts to zebrafish. Actually, it stops at zebrafish. There's now some emerging evidence, unpublished, that I know of, that it may occur in Drosophila, but only in certain tissues. So the difference between Drosophila and C. elegans is Drosophila also has lots of non-dividing cells, but the gut has some dividing cells. And in fact, Drosophila, as they age, develop a hyperplasia of the gut, which looks like a precancerous condition. And it looks like there may be senescence that occurs in the gut of Drosophila. So it may go way back to fruit flies, we're not sure. But what I can tell you is that the difference, say, between a mouse and a human is not enormous. It may be because we don't know enough about the species-specific comparisons, but when we look at that handful of markers that I told you about, they're conserved between at least those two species. So it's not clear that we will develop better therapies, well, we will need different therapies for, say, a human compared to a mouse. It's just too early days to be able to say for sure. There are lots of people now doing whole transcriptome characterization of mouse and human cells to try to get at just that question.
Sam Gutterman, I think big thing in the news is the Cancer Moonshot Project.
Yeah.
What aspect, and I haven't followed any of the details, what aspects that you've talked about are being addressed Or is it just let's try lots of approaches or better understanding? Or is there something specific that you've talked about that is being addressed?
My understanding of the cancer moonshot is the first goal is to try to identify what are called the driver mutations. So cancer genomes are a mess. And many of the mutations that cancer cells carry don't have a big consequence for the cancer cell. They're picked up because the genome is unstable and making lots of changes. But we know that there are certain driver mutations that are really driving that tumor to become more and more aggressive. So the first goal is to identify the driver mutations. Those driver mutations code for proteins, and the next goal is to develop drugs that target those proteins, so-called targeted therapy. And those drugs do work, but again, the problem is this constantly evolving genome, and eventually the cancer becomes resistant to those drugs. So the next step is now, let's throw a handful of drugs at these cancers so that there's so much pressure on the cancer, it doesn't have a chance to evolve these more resistant phenotypes. In addition, I do know that there are people very concerned about the whole tissue environment thing. So suppressing inflammation, suppressing that part of the tissue that's providing the soil for the cancers to grow, especially the metastases. Nowadays, people don't really die of a primary tumor, they tend to die of metastases. And so understanding the soil and changing the soil in a way to make it not so habitable for those metastatic cells. So I think it's a two-pronged approach. It is going to be a tough problem because cell division is the biggest risk factor for picking up new mutations. And so it's going to be tough. But I think the approach is sound and rational. It's just going to take a lot.
Anybody else? Yes, okay, we do have more questions.
Well, I'm trying to think. I'm Sue Sames, and I'm just trying to think of exactly what I want to ask. I appreciate your talk. I am like right in the middle of where it starts to. I'm 54, so I'm like right in the middle of where your risk for things start to greatly increase. So.
So you want the 25-word or less?
Yeah, I want the tweet of what I need to do next.
Eat your veggies, exercise. You choose your grandparents very well. That's the best advice I can give you.
Good to know. And just kind of, are there ways to learn more about what you're doing? Like what I really liked about your topic is that this is it's a very complicated topic, but this was very accessible. So what would you suggest if someone wanted to learn more but didn't want to get a PhD in biology?
Yeah, so there is you may have heard of the SENS Foundation, SENS Foundation, and they actually had a videotape, a bunch of lectures on aging. Again, mostly for a non-specialist audience, and they're accessible on YouTube. So you can just Google SENS Foundation and try to access their videos. And I at least people have to. I actually haven't seen them myself, but I was one of the ones that they taped, so I haven't even seen my own. But I do know that they put this together, and people have commented to me that they seem to be very accessible lectures. So that might be a good place to start.
Terrific, thank you.
Anna Rappaport, can you say any more about how this links to discussion about metformin and what the connection is?
Nir walked out.
I know.
Yeah, so metformin, Nir and I were just talking about this. It targets, he thinks the reason why it works is because it targets one of the enzymes in one of those pathways that we identified as driving the inflammatory secretions of senescent cells. So they might be related. We have not actually tested metformin on senescent cells, and we probably should. And I don't think that any of those patients who have been on metformin have been assayed for the presence of senescent cells. I mean, I think this might be an interesting epidemiological study to do.
We have one more.
David Watkins, Rothsay Life. I just want to ask a simple question, which is why do the mice with the CGV, I think it was, so the senescent cells are killed, why do they die eventually?
Oh, what do they die of?
Yes.
Yeah. So it's a tough question. I've asked that very question of Jan. Believe it or not, it is not uncommon for a veterinarian, the pathologist who's looking at these mice, they're doing necropsies on them to try to figure out what was wrong. And this also happens sometimes with people. Actually, it's not uncommon with very old people. On the death certificate, it says, heart stopped. I mean, so the mice, when they were necropsied, they had tumors, but they were smaller, and there was no evidence that the tumors had reached a stage where it was driving their death. But they did have tumors. They did have kidney failure. They did have some loss of cognitive function. But none of them were severe enough to really explain what the mouse died of. And so I think that needs to be determined. And it may not even be that easy. It could be that when you have multi-system decline, it's going to be very hard to point and say, this is what actually caused the heart to stop. So yeah, sorry, I can't give you a very clear answer. They do die. They do have evidence of tumors and multi-system decline, but it's hard to know what actually was the cause of death.
Thank you. So does the maximum lifespan does it have an intuitive explanation to you?
I'm sorry.
So the maximum lifespan, there being a limit,
yes.
Does that have an intuitive explanation in your mind?
How much of it is actually...
Oh, sorry, does it have an intuitive explanation?
An intuitive explanation?
Yes.
For what determines their maximum lifespan?
For the existence of an upper limit to maximum lifespan.
I'm just having trouble understanding the question.
Is there an intuitive explanation as to why even these mice with the senescent cells having been killed seem to just stop surviving after a certain point? I
mean, they're dying. Those mice without senescent cells are dying at about the same time as the mice with senescent cells are dying. So I think the key question is, what did evolution do to take a mouse which lives 3 to 4 years, 97% genetic identity to humans, and get this 30-fold difference in lifespan? We do not know the answer to that. I mean, there are lots of, the 3% of the genome that's different are all possibilities, but it could also be that you have to change so much in order to change maximum lifespan that the targets are maybe in the thousands. We absolutely do. This is something we do not understand, is what genetically defines lifespan in, say, 2 organisms that are reasonably closely related and yet have a huge difference in lifespan. We just don't know. I mean, look at the sharks, right? There are some sharks that live to be 40 or 50 years, and now we know there are some that live to be 400 years, and they're related. They're not that different genetically. I don't think we have a clue.
Thank you.
Sorry, Geordie Posthumus again. Could you say a word or two about what you know of mTOR's connection to senescence, if there is a special relationship, or how the mTOR pathway?
Yes. So we've studied that extensively. So the mTOR pathway, which is the target of rapamycin, is one of the major pathways that drives the secretion of inflammatory cytokines. And we've worked out the molecular biology pretty well. We pretty much understand how that works, how it is that mTOR, which is a kinase, how it eventually leads to the secretion of these different inflammatory molecules. It is not the only thing that drives the secretion, and that's part of the problem. As you know, rapamycin does extend lifespan in mice, but it's very small. It's, maybe 10.
So are you saying if you inhibit mTOR, then you reduce the excretion
That's
of the inflammatory
correct.
cytokines?
So
Okay.
rapamycin inhibits mTOR, and rapamycin extends lifespan and suppresses inflammatory cytokine secretion. So mTOR is pro-aging, and inhibiting mTOR is pro-longevity.
Sure. And then another quick related question is there anything interesting to say about autophagy as its
Yes.
connection to senescence?
Yes. So senescent cells they upregulate autophagy. We don't know if that's good or bad. As you know, autophagy is a process by which cells really they engulf their own organelles, and they can use it for good or for bad. So a little bit of autophagy is good because they can recycle nutrients, and it keeps the cells happy and healthy. Too much autophagy, the cells basically eat themselves to death, and they basically can't survive. And so we don't yet know whether the autophagy in senescent cells is good or bad. But we do know is that rapamycin upregulates autophagy, and the cells seem to be happy, but they're secreting less. So we don't know yet.
Actually, well, final question is, what's the lifespan of a senescent cell, and does that vary by tissue type?
So in culture, it's easy. It seems to be as long as the technician can feed those cultures, because they still need glucose and serum factors. In vivo, we don't know, because we have no way of lineage tracing those cells in vivo. And I know there are labs working on that. So,
final question, if I may. Do we know about the energy metabolism of senescent cells? The energy metabolism.
Yes, yes, we do.
preferred?
So they're definitely more glycolytic than, so they tend to, they produce a lot of lactate, you can see that, and they tend to rely more on glycolysis than on oxidative phosphorylation.
Great, thanks.
Hello, my question has to do with comparing the health of the animals that you have when you say that the animals, even if they don't live longer, they appear to be healthier, they have more fat, they jump around more, and so I was wondering if you have any way of, and I can see that if you have two different, a control and a treated animal, that you can see between those two sections that there are differences between how healthy they are. It appears the way that you described it, that this is just sort of a few different factors. And I wondered if you have developed any way of measuring the relative health of animals at a particular age or after they've been treated with these cells. I mean, I think that would be very interesting from the point of view of managing elderly, because if we could get these people to function better at a particular time, that would be a real plus.
So I just want to remind you, though, the experiment has not been done of taking an old mouse that is already in decline and asking whether you can reverse anything. So that experiment just has not yet been done. But as far as looking at health parameters in a mouse, the best way to do it is actually in some kind of cross-sectional study where you can then do pathology on multiple tissues. So that means you have to kill the mouse. And that has been done. So if you do cross-sectional studies, you can show that the liver seems to be functioning better, the kidneys are functioning better, the heart's functioning better, the heart has less fibrosis, et cetera, et cetera. Behavioral studies in mice are difficult because they're subject to interpretation. You can't ask a mouse, how does it feel? You can't ask a mouse, are you in pain, for example? I mean, we don't know. For example, in the case of the osteoarthritis, are they using that injured leg because the joint is working better, or is it because there's just less pain? They're very, very difficult questions to get at. And so there are people working on ways to try to distinguish between true measures of health in an intact animal, but most of the studies have been done cross-sectionally by just taking animals at different ages, sacrificing them, and doing pathology on the tissues. And we can't do that with people, obviously.
Okay, thank you.
Okay, one more question, and then we have to wrap things up.
Okay, this might be a longer question, but so you
have
may
5 minutes.
Just it could be a big topic, but glucose in diet, like specifically a ketogenic diet and senescent cells.
Yeah. So it's interesting. I have a new postdoc in the lab who worked on ketogenic diet in mice and rats. So it extends lifespan, the ketogenic diet. Again, mostly median lifespan, not so much maximum lifespan. And we just before he went on vacation, he said, I'd like to see if a ketogenic diet has any effect on senescent cells. So stay tuned. We just don't know. We just don't know.
Thank you.
Okay. Thank you, Dr. Campisi.